Hello. In this year of coronavirus, I've made a number of videos for my students, and I have playlists of them. The first playlist I made on coronavirus kind of focuses on the virus, its relationship to other viruses, its hosts, and then, the receptors that it binds to, its RNA, what it does inside cells, how it replicates its RNA, how it makes proteins, etc. And so that was the focus of the first playlist. Since then, I have made a second playlist, and this video here is summarizing the videos that I have here, and this will then focus on more the host angle of coronavirus infection. So what are human cells doing in coronavirus infections? And then some preliminary information about vaccines and tests for coronavirus. So this is meant to be a quick overview, and these individual videos perhaps go into some of these topics in greater detail if you are interested. And so first, let's talk about how many human cells can be involved in coronavirus infections and what the various roles are. First, there is the role of the cells which themselves are infected. So let's start off with the epithelial cells, which line respiratory passageways. Of a particular vulnerability would be the type 2 cells, which secrete surfactant. And so any cells in the body, and there are many, which produce the protein known as the ACE2 receptor, are capable then of allowing coronavirus to enter them. That receptor is here in green. And there are lots of different cell types in the body, and many express this ACE2 receptor. And so it is not just in the lung, which is obviously our first topic of conversation, given that this is a respiratory virus transmitted through respiratory droplets. But there's also expression of ACE2 in the small intestine, the gallbladder, the kidney, the testis, the heart, the colon. And what the effects are on these various cells is not yet known. But let's just discuss then what would happen inside a cell, which makes this ACE receptor. The virus spike proteins would attach to the receptor, and this would allow the virus to enter the cell. And I go into this in greater detail in the other playlist. Okay, let's say the cell is infected with virus and it might actually die. Well, it actually does have a role to play, because cells of the body wear kind of ID badges. All right, so it's important for cells to identify themselves. White blood cells are wandering around the circulatory system touching cells, and essentially they touch a cell and say, could I see some ID? Because if it's a body cell, it has ID and it can stay. If it's inappropriate, if it's a foreign invader, then the immune cell could attack it. These ID badges are what we call MHC proteins, or major histocompatibility proteins. So these epithelial cells, they have ID badges. But an interesting thing that these ID badges do is they grab a little something from inside the cell and then carry it to the outside of the cell. The little something in red is what's called a peptide. That is a fragment of a protein. So these ID badges have little fragments of proteins which were inside the cell. If an epithelial cell is infected by coronavirus may think it's hiding on the inside of the cell. But as it produces its red viral proteins, pieces of these could be then attached to those cell ID badges here and then taken to the cell surface. For simplicity's sake, I'm just going to have the spike protein here, but really that would be a peptide from the spike protein attached to an MHC protein. So now the virus thinks it's hiding, but actually part of the virus is being worn on the outside of the infected cell. And there are white blood cells which look for this. They are called cytotoxic T lymphocytes. They were formerly more likely given the nickname of killer T cells. So this cytotoxic T lymphocyte is looking for ID badges with foreign proteins, or foreign peptides attached to it. And so this epithelial cell, while a victim, it's reproducing the virus inside. Nevertheless, it is participating in the immune response. It can alert immune cells to the presence of coronavirus and help educate immune cells of what they need now to protect themselves against. All right, and so that is an important role. If this cell were to die, another thing that could happen is lots of proteins and viral RNA could be in the debris that could then be taken up by other cells, which I'll mention in a second. Why might this epithelial cell die? Well, virus inside a cell might lead to the death of the cell, but there are two other options. Cells have a self-destruct button. There is a pathway, and it's complex but very important, known as the apoptosis pathway or an apoptotic pathway, or more commonly called programmed cell death. All of our cells have a mechanism to destroy themselves. There is programmed cell death. And a cell that is infected by virus could trigger it on its own. A cell could realize things are going bad inside. I need to kill myself. Or this cytotoxic T lymphocyte could say, hey, here's a cell with the appropriate ID badge, but look, there's a foreign viral peptide attached to it. This cell needs to kill itself. And so programmed cell death can be initiated, apoptosis, by either the cell itself or triggered by the cytotoxic T lymphocyte. Cytotoxic T cells say, here's a cancer cell. They can secrete these little things called perforins, which make a hole. And now this cell has a hole in it, and its good stuff leaks out, bad stuff leaks in, and the cell is dead. Or the cell could, this cytotoxic T lymphocyte could just kind of press start on the pathway for programmed cell death. So even without putting a hole in the cell membrane, the cytotoxic T lymphocyte could now trigger through the appropriate receptor programmed cell death. Once the cell dies, now it's going to release inflammatory signals, which will attract other white blood cells to the area. These viral proteins can now be ingested by things like macrophages, dendritic cells, which can then participate in the immune response, which we'll discuss. So these epithelial cells aren't just victims. They actually then could be a part of the solution, although as I'll be pursuing presently, those inflammatory signals might actually contribute to part of the problem, but I'll get back to that. Okay, so that's one cell type. So now let's say in the lung, there are infected cells and there are epithelial cells dying. Well, there are immune cells already in the lung. There are dendritic cells, there are macrophages, and others. And then inflammatory signals released by dead cells can then attract more. So some of the proteins produced are actually things like monocyte chemotactic protein, which actually brings monocytes to the lungs. These things, the macrophages, monocytes, dendritic cells, and there are subclasses of each, they then eat the debris. All right, so they could eat a dead cell, a dying cell, maybe even a virally infected cell. They can absorb viral proteins, viral RNA, and things like that. And so let's say that somehow red viral proteins have ended up inside this macrophage. Well, it knows that this was debris absorbed from a wound site. So it then attaches it to an MHC protein and will wear this on its surface. Now, if you care, and say anatomy and physiology two students might care more, there are two different kinds of MHC proteins. This one's, I'm using the green color. These are the ones that immune cells like this macrophage would use to what's called present. An immune cell which touches a green MHC protein, an MHC2, doesn't say, here's an infected cell. It thinks, oh, here's an immune cell presenting or showing me a foreign peptide to beware of. And so this macrophage ingested part of the virus. All right, and so It ingests the foreign peptide, and it then becomes what's called an APC, an antigen presenting cell, which then wears this viral peptide on its cell surface as a signal for immune cells. Now, you may notice here's a lymphocyte, but it's not the red color of a cytotoxic lymphocyte. This is a T helper lymphocyte, or a helper T lymphocyte. So it is being stimulated by the green MHC proteins, the MHC2s. And it now knows, hey, our immune cells in the lungs, they're finding this foreign peptide. So the T helper cells, they're going to go and stimulate other cells like cytotoxic T cells, or like the B lymphocytes, which make antibodies. Now, this macrophage can stimulate the T helper cells and activate it. The T helper cells can activate macrophages as well. So these sparkles are these local hormones which can go back and forth. And so these macrophages and dendritic cells, they can help activate the lymphocytes. Now, not quite sure whether macrophages or dendritic cells could actually be infected by coronavirus. There's a lot which isn't currently known. They do express ACE2 receptors, so they could potentially become infected. As I will stress later, this is very important. Dendritic cells can actually take up RNA from the virus and actually make viral proteins inside the dendritic cell. So these could then attach to blue MHC proteins, the MHC1 proteins, which could attract the cytotoxic T lymphocytes. So these cells, even though they are what are called part of the innate immune system, they are stimulating these red cytotoxic T lymphocytes with their MHC1 proteins, and they can stimulate the T helper lymphocytes with their MHC2 proteins. And so they're playing a big role in activating the next part of the immune system. So these are part of what are called the innate immune system, which then have roles in activating the adaptive immune system. There are other cells of the innate immune system. I'm only going to focus on one, but I wouldn't be surprised if future publications point to roles that other immune cells are playing. I'd like to focus on the neutrophils. Neutrophils are the most common white blood cells in circulation. They are typically the first ones to get to an infected site, and they get there in great quantities. A lot of the pus at a wound is the neutrophils, which absorb microbes, and things which have bound antibodies and then blow themselves up. So neutrophils are certainly going to encounter coronavirus infected cells, because when there's inflammation, they get there first. And neutrophils perform phagocytosis of things. And so, they might ingest coronavirus, but on its own, maybe. Once again, I'm looking forward to the publication of greater understanding of this. But the one thing that they would certainly do is, once antibodies start sticking to coronavirus, that is a signal for neutrophils that say, hey, antibodies have stuck to this foreign thing. We definitely want to absorb this. And so neutrophils will be important in clearing coronavirus, especially once antibodies have bound to it. So in that, they are part of the solution. Actually, there's a name for this. When antibodies stick to something and this encourages white blood cells to perform phagocytosis, that is what is known as opsonization. So just antibodies is one of the ways that they work. They act as opsonins, which help the phagocytes recognize it and ingest those. So neutrophils do that. Now, something that's coming to light is, while neutrophils typically go up during an infection, and they certainly go up when inflammation is heightened, neutrophil numbers go up potentially a great deal during coronavirus infections. And in those who suffer the worst outcomes in coronavirus, they're especially elevated. And so that's one of the symptoms of coronavirus infections at their worst, a heightened neutrophil count. One of the things that we see in this coronavirus infection when they go extreme is lots of inflammatory signals. Now, inflammation is part of an infection, and it's good. It attracts white blood cells to the damaged area. But too much is a problem. And these local hormones promoting inflammation, known as cytokines, there are so many which can be produced in some people's infections that it's what's called a cytokine storm. Neutrophils may have a role in this. Those dead epithelial cells may have a role in this. The dying lymphocytes may have a role in this, as I'll get to. But for whatever reason, neutrophil counts go up. So you could count the number of neutrophils to lymphocytes and look at the ratio. Healthy people, 95% of the time, have a ratio less than 3.5. So for every lymphocyte in your blood right now, you may have, maybe 3 or maybe 2 neutrophils. So neutrophils outnumber lymphocytes in healthy blood by a factor of, 2, 3 or something. In almost everybody, it's less than 3.5. Now, as we look at coronavirus patients, this number tends to go up. The more that it goes up, the worse this person's prognosis is. And so one of the important diagnostics is a neutrophil to lymphocyte ratio. So let's say your neutrophil count was maybe 6 to 9 times that of lymphocytes. That's showing that there's a sign of stress here, but still this isn't as bad as it could get. NLR, once again, neutrophil to lymphocyte ratios, over 18 are indicative of severe stress. And I've read that, in those coronavirus patients struggling for their lives, it could actually approach 100. So while a normal person maybe has 2 or 3 of these neutrophils for each of these lymphocytes, a person who is in intensive care, I mean, it could be 50, it could be 80, it might even approach 100 of these neutrophils for every one of these, in part because the neutrophil number is going up, in part because the lymphocyte number is going down. Now, this is crazy important because these cells, they talk to each other by making signals. Neutrophils make signals that, help lymphocytes. Lymphocytes make signals which help neutrophils. And once we see this imbalance where, the neutrophil number is just too high and the lymphocyte number is just too low, and maybe you have all of these epithelial cells which are dying and these monocytes which are responding to the infection, you can have all of these signals and thus an imbalance. So as I'll see and I'll get back to, all of these proteins are local hormones, signals, which a coronavirus patient typically expresses more of than a healthy person. And all of these signals are ones which are overproduced in coronavirus patients who require intensive care compared to those who have a mild infection. And so the immune system is complex with all of these cell types which talk to each other, and there's a balance that we maintain in health. But one of the problems with coronavirus is an imbalance is reached, an imbalance in the ratio of cells, an imbalance in these local hormones known as cytokines, which then results in adverse effects. So that then is an important concept. Coronavirus is bad. The virus is bad. It causes all of these infections. But if you were then to ask, why might someone die of a coronavirus infection? Why might someone require intensive care? What seems to be true is that to a large degree, it's not the virus which is causing that severe effect, at least not directly. It's our body's response to it. This cytokine storm that the virus triggers then causes imbalances all over the body. These signals from immune cells are not only affecting other immune cells, they're affecting all sorts of metabolic processes. And so if there is an imbalance where we are overproducing inflammatory signals, this is going to affect hormones, it's going to affect blood pressure, it's going to affect muscle contraction. It affects so many things. And so this balance is important because some people get coronavirus and it's a mild infection. They may be asymptomatic, they may recover fully with very little discomfort. Some people recover after a month in intensive care and require, very intense intervention to save them. And then tragically, at this point, hundreds of thousands of people in the United States have lost their lives. And so the imbalance in our body's response is a major player in this, perhaps more directly related than the virus itself. So once again, all of these are signals, cytokines, typically pro-inflammatory, which are overproduced in coronavirus patients. And then there are signals which are overproduced in coronavirus patients with severe infections requiring intensive care compared to those with more mild infections. And this cytokine storm then is the problem. So if one were to list, if you have a cytokine storm, if you overproduce these signals. Now, the responses can vary from person to person, so I'm not saying that all of these are constant in every patient, but things that one can see are headache, dizziness, confusion, tremor, hypoxia, pulmonary edema, rapid and shallow breathing, changes in liver's enzyme production, increase in the gallbladder size, diarrhea, blood in the stool, abdominal pain, rapid heart rate, arrhythmia, cardiomyopathy, possible heart failure, an enlarged spleen, abnormal kidney filtration, renal failure, nausea, vomiting, fever, fatigue. If one asks, what is it about the coronavirus, which has killed hundreds of thousands of people in the United States so far? A large part of the answer is the body's response to the imbalance in our immune response and this cytokine storm. So once again, while we would probably rush to focus on those parts of the immune system which are making antibodies and the part of the long-term immunity, and that's certainly appropriate, some of these innate factors and the inflammatory signals that they are making, that is critical as well. Now, I mentioned dendritic cells, which are antigen-presenting cells like macrophages. Now, they may actually have a key role in the coronavirus vaccine. So here are videos which kind of repeat some of what I had, earlier said as kind of a precursor to the discussion of the vaccine. Once again, I'm just going to quickly go through this coronavirus. These cells called dendritic cells, they probably are presenting coronavirus peptides with their MHC1 proteins, and they are probably presenting them on their MHC2 proteins, activating both cytotoxic T lymphocytes and helper T lymphocytes. One thing that a scientific paper that I had read, there is another receptor which dendritic cells possess, which doesn't allow coronavirus to enter the dendritic cell, but it actually might allow a dendritic cell to transport the virus to another cell like an epithelial cell. So, dendritic cells are important to study. One of their receptors, the DC sign receptor, might actually have a role in worsening in infections. So there's this DC specific intercellular adhesion molecule, which seems to have another role and might actually make these dendritic cells part of the problem. Okay, so I go into them a little more detail and how they take up the RNA, but that is so that that this might be extremely relevant when talking about the vaccines. So I'll get back to that later. One other thing, and I'll mention with the vaccine, dendritic cells are worth focusing on for one huge reason. A lot of cells activate lymphocytes and present to lymphocytes in one place, like in the lungs or at a site of infection. But there is something that dendritic cells can do that they are unique in doing. And there's only some of the dendritic cells, there are different classes. What they can do is, and like I said, there's many classes. I have more videos, but that kind of goes beyond this. Look at all these kinds of dendritic cells, all right? What some of them can do after they, say, interact with a coronavirus, they can move. They, instead of waiting at the infection site for white blood, for lymphocytes to come to them, they can go to a lymphatic vessel, and they can then go to places where lymphocytes mature. So they can help to educate the immune system about the presence of coronavirus by actually going from, say, the lungs where the infection is to, say, a lymph node. So here is a dendritic cell precursor traveling to, say, a lymph node, where it now gets to teach the lymphocytes, hey, here's something, an infectious agent in the lungs, we need to start our response to it. That actually may be key in why the vaccines have proven so successful so far. So for example, it may be in the lymph node then that the cells which were educated then stimulate B lymphocytes to make antibodies that then go into the blood, which then help our response to the infection. So dendritic cells and their ability to travel may be key. The type of immune cell which makes antibodies is what is known as a B lymphocyte. So, and in general, lymphocytes start off in the bone marrow. The ones which finish their development in the bone marrow are called B lymphocytes. These are the ones which make antibodies which can be secreted. The ones which go to the thymus and mature there are called the T lymphocytes. So here you see lymphocytes releasing antibodies. These are the B lymphocytes. And if we have any immunity towards coronavirus infections, either after a vaccine or after becoming infected, it is because we have the ability to make antibodies against coronavirus and its protein. So clearly, B lymphocytes are important to discuss. Now, while we typically focus on B lymphocytes and we talk about them as the makers of antibodies, they are. Also, one other side note, they are also antigen presenting cells. While that has been known for at least a bit of time, its significance isn't fully appreciated, at least not in what I've read. So does that mean B lymphocytes are ingesting full coronavirus and presenting it to T cells? Are they taking up coronavirus proteins and presenting them to T cells? Is their role not important in that? Like, is it more important in developing tolerance, say, in fetal development and early birth, but not so important afterwards? I wish I knew the answer to that. But B lymphocytes are antigen presenting cells. So it could actually be that they are presenting to T helper cells and signaling them of coronavirus in infections. So there's that potential. And once again, that isn't understood as well as we would probably like. And once again, so a B cell potentially then could either bind either a spike protein or maybe even a whole virus, rip it into pieces, and then attach peptides from viral proteins to MHC2 proteins, and then signal T helper cells to activate those. And then T helper cells could then stimulate this cell to make antibodies, which then get released into the blood against that, against coronavirus. Now, what can an antibody do? All right. Well, antibodies do a lot of things. So one of the things that they could do is block the interaction between the spike protein and the ACE2 receptor. So this virus wants to bind to this receptor so it can enter a new host cell. But if it is covered with antibodies, then that might block it. So this might neutralize the virus, making it unable to enter cells. So once again, one of the main things that antibodies do is if this, foreign something has to fit here, once it's got antibodies stuck to it, it can't fit there anymore. And so that is one of many ways that antibodies can be effective. Antibodies are these wonderful things that have multiple ways of being active. Another way, as already mentioned, is once an antibody sticks to things, this will then signal cells of the innate immune system like neutrophils or macrophages, et cetera, to perform phagocytosis. So a neutrophil may not be able to recognize coronavirus. I mean, it might bump into coronavirus and not know that it's something to ingest, whereas if antibodies are stuck to it, then it might kind of act as a handle, which allows it to more easily bring this in. Now, this comes up multiple times, when talking about testing, et cetera. Here's an antibody, and this top part is the part that would stick to coronavirus. We can change out the bottom part, though. Okay, so this constant heavy chain can be changed, and I'm going to make it have different colors. Let's say the blue base here is what we call IgM. So if an antibody has the blue base, we would call it IgM in this analogy. But we could change the blue base out and put a different color in, maybe red, in which case we would call it IgG, or yellow, and call it IgA. The top part would still bind to coronavirus, but the bottom part now makes a difference. Okay, so that'll pop up, like for example, IgM, whenever you secrete it from the cell, you secrete it as a group of 5. IgA is secreted from the body. IgG tends to be in the blood. So the base tends to be important. So here, notice you've got 5 antibodies stuck together. That's IgM. All right, whereas IgG, each Y-shaped antibody would be different. As we'll see later, talking about tests, IgM tends to be produced early in infection, peaking in its production in the first month and declining afterwards, whereas others act longer over the longer term. Now you can see how 5 stuck together could be an advantage, because let's say this antibody sticks to this viral particle here. Well, now we could have a second portion of IgM stick to a second coronavirus particle, a third, et cetera. And so now these coronaviruses, we've tied them together. Like imagine I was trying to rob a bank, with some friends, and all of a sudden you could tie 5 of us together. We, want to commit a crime, but we won't be very effective, if we're all, bound together, we're less mobile. So IgM being secreted as a pentamer with 5 antibodies stuck together, it's good at what's called agglutination, where it will stick these together. They're also heavier now. This will attract other, white blood cells to ingest them, like macrophages, arguably less likely to be spread in an air droplet now that it's heavier. So IgM is certainly important. And as you can see here, if you look at days after, the first symptoms of coronavirus, IgM tends to peak early, all right? And so here we are, we're in our first month, and after that, the production goes down. And that's the opposite of what's called IgG. IgG is a different antibody. It's a red base, let's say, instead of the blue base. So the coronavirus binding part is still the same, but these are secreted singly as opposed to being in a pentamer. So they are produced later in the infection, and their secretion would be prolonged for months afterwards, and greatly increase if there's a second exposure. And so if you were to look at, like if someone had an antibody test and you didn't see any IgM, but you saw IgG, you might think, oh, you were infected a long time ago. All right, so yes, you were exposed to coronavirus, but this isn't a recent infection. Now I'm going to go on. There are other types of antibodies. So there's IgA. Most people don't focus on this. The antibody tests don't focus on this, but it's important. IgA is secreted from the body, like in the lung fluid. So obviously, since coronavirus is a respiratory illness, the lung fluid would obviously be important. I've been focusing on the antibodies against the red spike proteins, but coronavirus has other proteins like the N protein here in blue, and lots of antibodies form against the N protein as well. I can mention this again later, but antibodies are specific, all right? They specifically bind to one antigen. But what happens if you have two antigens which are similar? So here's just an interesting thing. COVID-19 is caused by SARS coronavirus 2, but there was a SARS coronavirus 1, a relative. Most of the antibodies against SARS-1 don't really work against SARS-2. Most, but some do. And if you notice, there are some of these coronaviruses which are actually causes of the common cold. So some coronavirus are serious infections, but there are coronaviruses which you may have been exposed to already. And the fact that you've already been exposed to coronaviruses mean that some people trigger a positive antibody test, but they haven't been exposed to COVID-19. They've been exposed to one of its relatives that caused the common cold. So you just have to be careful. And obviously, if we want to interpret coronavirus test results, the fact that antibodies are specific, but there is a little bit of overlap, it is just one a fact to keep in mind. Now, in severe coronavirus infections, lymphocyte number drops, and that includes the B lymphocytes, which are making the antibodies. All right, and so when we talk about B lymphocytes and their roles, the fact that in those coronavirus patients who are in intensive care, we may see a decrease in the B lymphocytes that is obviously a potential problem as well. So B lymphocytes decrease, NK cells decrease. Those are not quite lymphocytes, but they're similar to lymphocytes. So that's a type of immune cell we like to discuss as well. And T lymphocytes decrease. Now, T lymphocytes are complicated, and just to be honest, there's different kinds. There are the cytotoxic T lymphocytes. These can recognize infected cells and then cause the infected cell to die. They can trigger apoptosis. They can secrete perforins, which put a hole in the cell membrane and they kill the cell. So cytotoxic T lymphocytes kill infected cells through two different mechanisms. So this is a type of T lymphocyte which is involved in coronavirus. Once again, T lymphocytes mature in the thymus as opposed to B lymphocytes, which mature in the bone marrow. So here's a cytotoxic T lymphocyte. Once again, it just put a hole in an infected cell or a cancer cell, and that kills it. Then there are the helper T lymphocytes, the T helper lymphocytes. They are important in giving permission to other parts of the immune system to act. So B lymphocytes don't make much in the way of antibodies until T helper cells give them permission. Cytotoxic T lymphocytes don't really go around killing cells until T helper cells give them permission. And so these lymphocytes are critical because many other parts of the lymphatic system can't act without their permission. So they are themselves stimulated by those antigen presenting cells like dendritic cells and macrophages, and then they go around stimulating other parts of the immune system. So they are very important. There are also other types of T lymphocytes. Some are called Treg cells, which help maintain a balance in, say, pro-inflammatory states, anti-inflammatory states, et cetera. So here, I'm sorry, you can see here's a B lymphocyte and here's the T helper cell, which is then stimulating it. And after the T helper cell responds, or after the T helper cell simulates it, then the B lymphocyte makes lots more antibodies, and the response with the help of the T helpers is much greater than it would be without it. And so other T lymphocytes and T lymphocytes in general are very important in, once again, balancing the signals which are coming from all over the place, because as you can see, there's all these different kinds of cells that are interacting with each other. So a balance in their interactions is important. Sometimes we characterize some of the interactions as like Th1 and Th1 T helper cells versus Th2 T helper cells, which may secrete, say, pro-inflammatory signals or anti-inflammatory signals. All right, so bottom line, T lymphocytes, lots of roles in this, which is why it's such a problem that in severe coronavirus infections, their numbers really drop. Their numbers really drop in AIDS, and their numbers really drop in coronavirus sometimes. People have mild infections, no, but in severe infections, yes. And that's why one of the reasons that NLR ratio becomes so distorted. The neutrophil numbers are going up and the T lymphocyte numbers are going down. Does coronavirus infect and kill T lymphocytes? Possible, but not yet known. So AIDS does. HIV kills T lymphocytes. Why the numbers are dropping so much in coronavirus, not entirely clear. Does coronavirus infect and kill T lymphocytes? Does it promote their apoptosis? Does it destroy lymphatic tissue where they are made, etc.? Not entirely clear. But once again, getting back to the NLR, a problem with coronavirus is that some people experience no symptoms and that other people die. And obviously, if you're a healthcare provider, distinguishing between patients who need immediate intervention versus those who don't and who can recover at home just by quarantining, that's important. All right, some people stay at home and quarantine. Other people, you need an ICU bed. How do you distinguish between those patients? One of the ways is by one of the best diagnostics is to perform that neutrophil to lymphocyte ratio. All right, normally it's around 3. This patient here might experience, only mild symptoms. All right, but if someone has a much higher NLR, that is a warning sign with the idea that this person would need much closer medical scrutiny, may need an ICU bed, etc. So this NLR is a very important diagnostic to estimate the severity of coronavirus infections. And so I have a little video that just repeats what I've just said about the NLR. Okay, so I'd like to shift gears into the second of the 3 topics and now talk about the vaccine. So, the first thing was just going through, the diverse cells of the body and diverse cells in the immune system and how coronavirus is thought to affect all of these. And I'm making this just in January of 2021. I'm sure that far more will be known in the immediate future, but I'd like my students to try to at least be exposed to what was at this moment up to date information. So vaccines, how do they work, and how does the coronavirus vaccine works? Well, the coronavirus vaccine is an RNA vaccine. Now, RNA is already involved because coronavirus is an RNA virus. RNA is its genetic code, and when it infects a cell, it causes the release of coronavirus RNA into the host cell. And then the host cell makes coronavirus proteins from the coronavirus RNA. So here's a human ribosome, which reads this thinking that it's a human RNA message and makes protein thinking that this is a human RNA, a human protein, but it's instead the viral protein. The virus is producing all of the proteins it needs to propagate itself. So inside host cells, viruses are using RNA to propagate themselves because the RNA has the instructions that coronavirus RNA codes for the red spike proteins. That's important. I'll focus on that. But also then other, proteins as well. There's a bunch of proteins in coronavirus, not just the red spike proteins, although once again, that is what I'll focus on because it is the red spike protein, which is binding to the human ACE2 receptor. And this is how the virus enters host cells. Now, if we could make antibodies against the spike proteins, that could largely block the viral particles from entering host cells. It would block their ability to bind to the ACE2 proteins, plus because it's on the surface. Then the antibodies could bind to it, and this could attract neutrophils. Okay. So vaccines, as I will say, there are traditional vaccines, and the coronavirus is something brand new, the coronavirus vaccine. In a traditional vaccine, you're going to inject something into an animal. Let's use this lab rat. So you could use virus. Now, how would you cure a disease by injecting virus? Well, it could be a strain of the disease which is mutant and therefore not capable of causing infection. So if we injected this mouse with a non-virulent strain, the mouse's immune system could learn to recognize coronavirus and thus make antibodies against it. However, if this mutant virus mutates a second time, it is conceivable that it goes back to the original form, which can cause disease. So it is possible to get a bad batch of this traditional vaccine, which actually causes disease. Sometimes you would heat the pathogen that would make it non-virulent. But if the vaccine wasn't prepared carefully, if there wasn't, quality control, then maybe once again, you could have a bad batch of vaccine, which would either cause the disease or not protect against the disease. Also, you have to make virus. If you're going to be injecting virus, you have to make it, all right, that it can be packaged in here and do quality control, and that would take much longer. So if the vaccines against coronavirus were traditional vaccines, they would not have been available by the end of 2020. They would have just taken much longer to develop. All right, so traditional vaccines in this sense, if they're going to inject a non-virulent strain, they've got disadvantages and they would take longer to develop. Another way of making a traditional vaccine is if you could mass produce, say, the spike protein. If you could inject the spike protein into the host, now the immune system of the mouse would learn to recognize the viral spike protein and make antibodies against it. And so that would be a way of training the immune system. Now, you have to make this red protein if there's a problem in your production, and there's problems with anything. Then you would potentially have a batch of vaccine that didn't work. Maybe the red protein was denatured. It had an abnormal shape. It wouldn't work normally. Plus, you'd have to make the protein, purify it, maintain it. Once again, it would take longer, and you'd need more quality controls, more testing. So coronavirus first started infecting people in the United States in 2020. By the end of 2020, there were vaccines. If these vaccines were traditional vaccines, they would not have been prepared in this amount of time. Traditional vaccines take longer. The coronavirus vaccines, the first ones approved in 2020, I'm sure at some point, I'm sure there are other ones, they were not traditional vaccines. So now let's talk about this non-traditional coronavirus vaccine. The coronavirus vaccine involves viral RNA, which is being injected. Now, the virus does that. All right, so the virus injects its RNA, and then the human ribosome then follows the instructions. It puts amino acids together the way the virus tells it to, and it makes viral proteins. This is how the virus makes viral proteins inside human cells. The coronavirus vaccine does the same. It introduces viral RNA into human cells, and the human cells then make viral protein. So here's that red spike protein being made as this human ribosome inside the human cell, follows the instructions of the viral RNA. Now, this sounds infectious. It certainly doesn't sound like a vaccine. How is this safe? And the answer is because the vaccine doesn't include all of the virus's RNA, just one tiny little piece. So the virus makes all of its proteins inside the host cell, but the vaccine only has genes, only has one gene for the spike protein. Let's say here's that little red portion. So, it's like a car. Wheels are obviously important to the car, but if you just have a wheel, you don't have a car. The wheels are an important part, but you need all of the other parts as well. This is just a part of the coronavirus, the spike protein. It by itself isn't capable of infecting a cell. It's just a protein. It's just a part. And so the RNA vaccine has just the instructions on how to make this one viral protein, not all of the viral proteins. If you've never heard of this, that's because this, was thought to be impossible. The first time it was demonstrated to be effective, and most people felt it wouldn't work, was 1994, which is relatively recently, and it wasn't approved for human use until much more recently. The beauty of this is that it is the host cells which are making the red spike proteins. Therefore, you don't have to make it in a factory. You don't have to do quality controls. You don't have to purify it. And since you're just making the spike protein and not the entire pathogen, you're limiting the chance of an infection. So this is superior to the traditional vaccines. Plus, it's relatively easy to generate the sequence for that one protein. This is the reason that the coronavirus vaccine was developed in record time, because it was a brand new type of vaccine which could be developed at a faster pace as opposed to the traditional vaccines which would take much longer. So once again, the virus has lots of RNA, but the vaccine only has this red portion for the spike protein, not the rest of it. Okay. Now, let's just talk about RNA viruses in general. The first RNA viruses demonstrated used what's called naked RNA, just plain old RNA. And that was shown to be effective. Somehow, the red RNA was ending up inside host cells. The host cells were making protein from it. And so somehow, if you just inject RNA that ends up inside cells, they make the red virus proteins, and this can sensitize the immune system. Naked RNA works. Now, it's not as effective as it could possibly be, but I'll get back to that in a second. How could naked RNA work? Well, if, say, this dendritic cell is infected by coronavirus, the red RNA would cause the cell to make red protein, and this could stimulate, the cytotoxic T lymphocyte as I just, described. So, there's that. So naked RNA, could work that way. But how do cells take up RNA? That's usually not their thing, right? So cells usually don't take up naked RNA. I mean, we don't think so. I mean, that's makes it easy for infection. So how is this possible? And then going back to 1994, that's why a lot of people didn't think it would work. Why would a cell take up RNA? And so this, at the time I'm making this video, this seems to be part of the answer. Maybe it's not the whole answer, but this seems to be part of it. That seems to be part of a dendritic cell's job. If it goes to the site of an infection where there's dead cells everywhere, including maybe dead virus RNA, the dendritic cell absorbs that on purpose, all right? So it takes up the red viral DNA on purpose, makes viral proteins on purpose so that it can now present, all right, to immune cells, okay? Why would it want to do this? Well, if you want your immune system to be signaled by these antigen presenting cells against viruses which infect human cells, remember that not every virus infects immune cells. All right, so there's viruses which only infect liver cells. There are viruses that only infect brain cells, et cetera. So the idea that dendritic cells on purpose are trying to grab viral RNA, make viral proteins, that may be part of their job to try to signal immune cells of, beware of this virus, even though it's not a virus which would normally infect a dendritic cell. So dendritic cells, that may be significant in the vaccine, that they actually seek out RNA to take up. Okay. Now, I'll get back to that briefly. There are other things that one can do. So it was shown that even though naked RNA works, could it work better? And it turns out it can. If you just apply electric current to the area, that will cause cells to take up this RNA at a higher rate. This was demonstrated for the first time in 2009. Now, this can actually be done with cells. So if you want bacteria to take up new DNA, if you want plant cells to take up new DNA, you can actually put it in a little cuvette. You can actually, have it occur in liquid, in a cell suspension, in a little, apparatus known as an electroporator. This process, electroporation, is a way of increasing the amount of RNA which is taken up. You could actually do it on this mouse if you applied electric current at the site of the injection. Now, that's not what the current coronavirus vaccines used, but because this is a brand new way of vaccinating, this is coming. All right, so RNA vaccines whose effectiveness is increased with a small electric current, this electric current will make pores in cells which allow them then to take up this RNA. This is coming. Vaccine development is forever changed now because of this new type of vaccine. So worth remembering that you can increase the effectiveness if you don't use naked RNA alone. Another way of increasing the effectiveness of RNA vaccines is to now use a lipid nanoparticle around the RNA. This stabilizes the RNA, that's important as I'll get to, but also it might allow the lipid to fuse with a cell membrane. This is brand new. So lipid nanoparticles for drug delivery was first approved in 2018. All right, so the coronavirus vaccines use RNA to vaccinate. That was first approved for human use in the year 2020. But they are using lipids around the RNA to increase the effectiveness. That was first approved for human use in 2018. So, this is, a brand new way of developing and delivering vaccines. I'll mention RNases again in just a second. One other thing which, once again is groundbreaking is which lipids you use might allow you to target specific cells of the body. Cells have different receptors. And so depending on what you put in the coat, that might allow you to actually deliver RNA to some cell types in the body rather than, generally throughout the body. Both of the two first vaccines, the Pfizer and the Moderna vaccines approved in 2020, were not only RNA vaccines, but also RNA vaccines using lipid nanoparticles, which makes the delivery of the vaccine more effective. All right, they both need to be kept very cold. I'll talk about why in just for just in just a second. Okay. And so corona vaccines will forever be different because of these novel vaccine aspects, first used in 2018, 2020. All right. Now, because this is so new, obviously, we don't understand everything we could about it, and lots of people would have questions. So, for example, can the coronavirus vaccine cause a coronavirus infection? I mean, wasn't it true that some people would actually get polio after getting the polio vaccine? So the answer is no. The reason is because viruses need to make a lot of proteins, for the capsid, for incorporating themselves into a cell, etc. But this is not all of the virus. All right, so here I'm just saying the traditional vaccines, they have the ability to cause infection on rare occasions if something goes wrong, but that is not true of this vaccine, because it's not making the whole coronavirus. It's just making the red spike protein. So I talk about this in my other playlist about coronavirus. Coronavirus makes a lot of different proteins. All right, so it has a big genome. All right, and all of these little pieces, these are little proteins which are made by the coronavirus particle, which are needed for its infection. So coronavirus can be effective because it makes all of these different components. The spike protein is just one of these that will be encoded by this portion of the virus's genome. So this is why you can't get coronavirus by just making the coronavirus spike protein. It is just one component of many. So inside a coronavirus particle, you have lots of genes. The red gene for the spike protein, that is just one piece, like just the wheels of a car, going back to my previous example. And so when you get a vaccine, you are not making the whole virus, you are just making the red spike proteins, and that is not enough to get sick. But it is enough to train your immune system. If you can make antibodies against the red spike proteins, then these antibodies can bind to coronavirus and then prevent them from attaching to the receptors, etc. Now, a second question one might ask is, why do these vaccines need to be kept at such cold temperatures? And the main answer is because we are dealing with RNA as opposed to DNA. RNA is less stable. Now, if we were to just use DNA as a reference, why is DNA so much more stable than RNA? Well, it's wrapped around these histone proteins in the nucleus. It has that adds to stability. It has a double helix that adds to stability. The nucleus that keeps the DNA doesn't have many enzymes that break DNA down. So while there are enzymes called DNases, which degrade DNA, you don't really find them in the nucleus. In the cytoplasm, you find lots of RNases, right? So RNases are much more widespread, which can destroy RNA. DNases, you don't find them to the same degree. The DNA code's a bit more stable. There's that reason. But a main region, so the RNases are enzymes which eat RNA, and they're very common. That's one big reason. Here's the second big reason. So just a little bit of chemistry. A DNA nucleotide uses a sugar called deoxyribose, while RNA uses a sugar known as ribose. Ribose has an oxygen at the number 2 carbon. So yes, we number them. My video goes through it if you want, but I won't focus on that now. The number 2 carbon has an OH group, whereas in DNA, deoxyribose just has an H. Why does that matter? Because OHs are reactive, and just plain old H's, they tend not to be. So a fatty acid, the fat part, has just carbons and hydrogens. It's inert. It doesn't really react much. But the acid part, the N, it has an OH, which is very reactive. So OH is reactive, missing the OH, not so much. So DNA, just the sugars in the DNA nucleotides are not as reactive, while in RNA they are. And that matters because let's imagine you have a chain, all right? Once again, here I'm just numbering my carbons. Here in the chain, this is ribose. Notice there's an OH here. Now, what I want, if I want this RNA to stay as a chain, there's sugar phosphate, sugar phosphate, sugar phosphate, sugar phosphate, sugar phosphate. I'd like the sugars to stay stuck to phosphates, and I'd like this phosphate to stick to sugar on this nucleotide, and then with its other end stick to a different sugar on a different nucleotide. That's what keeps them in a chain. But what's going to happen is that this phosphate, instead of reacting here, it's going to react here, so that this phosphate, instead of being stuck to sugar number 1 and sugar number 2, it's now going to stick to sugar number 1 at 2 different places. This breaks the chain. So RNA, the chain gets broken more commonly because of a bond that can form here. So this then is the reason that the coronavirus vaccine needs to be kept so cold. It's a new kind of a vaccine using RNA, and RNA is just less stable for the reasons that I've just mentioned, that the chains can break apart, plus there's the enzymes which degrade RNA are more widespread than those that would degrade, say, DNA in a comparison. Now, the first 2 vaccines were both RNA vaccines, but they differed in how cold they needed to be kept. So if both of them are RNA vaccines, why does one need a colder temperature than the other? And I don't know. And one of the reasons I don't know is those who are making vaccines, which are obviously now being manufactured, they're going to be quite lucrative. There are some, say, industry secrets. And so the exact sequence of the RNA used by different companies might affect what's called the secondary structure of the RNA. RNA molecules can fold on themselves a little bit, and the degree to which that happens would vary depending on the specific sequence. One might be more stable than another, so that might affect this. Also, the specific lipids used in the lipid nanoparticles, they could vary. Which ones are being used? Once again, I don't know, in part because this is, in part a proprietary secret where, manufacturers, are guarding their own, patent, stuff and procedures. And so differences in the RNA sequence, which affect structure, differences in the nano particles, which are stabilizing the RNA, this could then play roles in that. So this is exciting for lots of reasons, not just for coronavirus, but vaccine production will probably be different forever from this point on because of this, groundbreaking novel way of, looking at vaccines. A question I mentioned, kind of alluded to earlier. Let's say a new mutant strain of virus develops. This is early January 2021, and there is concern that a new strain of the virus first discovered in the United Kingdom and another strain first discovered in South Africa have spread throughout the world. If you make a vaccine against one, what are the odds that it will be effective against others? That's a good question. As I had mentioned, that the antibodies against SARS coronavirus 1 sometimes, but typically are not effective against SARS coronavirus 2. So might a new strain mean that the vaccine is no longer effective? And the answer is only time will tell. But to be honest, no. So that is not a huge concern. And let me explain why, if I can just get to the right place of the video. Um, there is a difference, if you really wanted to study this, in what's called monoclonal antibodies and polyclonal antibodies. So let's just say, here we inject, say, a sheep with, let's say, coronavirus proteins. And we start then making antibodies against the coronavirus proteins. The truth of the matter is a protein's a pretty darn big thing. And one of the B lymphocytes might make antibodies against this piece of it over here. This one might make antibodies against this piece over here, or this piece over here, or this. So now in the sheep, yes, you're making antibodies against the coronavirus protein, but you're probably making different antibodies against different parts. If you're into antibody production, the term is epitope. And polyclonal antibodies are now making antibodies against multiple epitopes from multiple B cell lines. Now, what that means here is, let's say the UK variant, I'm making this up, is more transmissible. Why? Well, obviously there's a change, but it might just be a change over here. Might that affect, say, the red antibody, where now a change in the protein structure might make the red antibody less useful? It's possible. But antibodies are binding to more spots than just that one little region. And so while there's a huge difference between SARS coronavirus 1 and SARS coronavirus 2, just kind of like there's a huge difference between like a house cat and a lion. They're related, they're both cats, but they're distantly related. So there's a lot of differences between them. There's less of a difference between, say, this breed of house cat and that breed of house cat, which are now, almost entirely similar, just minor changes. So while only time will tell, I mean, science can only make conclusions based on observations. And the question, will the vaccine work against multiple strains of virus, including new mutant strains, which haven't, even arisen yet? In some degree, we don't know. But at least in the short term, the answer is yes, the vaccine should still work, because there's going to be different antibodies going after different parts of, say, the spike protein. And if a new mutant has, say, a slightly different part in one, that might not make a huge difference. In this video, I just kind of recap the dendritic cells. But once again, the idea that dendritic cells, it's part of their job to take up foreign RNA from, say, potentially a virus and express it as a way of signaling immune cells, that this is something that you could look out for. That's actually exciting. That's something that's not really appreciated about dendritic cells, if that's true. And now the idea that then they could then travel. All right, that part of their job is to say, even though this virus doesn't infect dendritic cells, I'm going to simulate an infection by making viral proteins. And not only will I wear them and present them to lymphocytes, I'll actually travel to lymph nodes where the lymphocytes are being produced to signal them there. That's actually kind of exciting. One last point before I get into different tests is there's plenty of other things, including, for example, the idea that vitamin D deficiencies might actually make coronavirus worse. Vitamin D is something that we can eat. If we eat a lot of fish, if we eat a lot of mushrooms which have been grown in the sun, especially if they're dried, or if we're ingesting food which has been fortified with vitamin D, like milk. But for most of us, most of the vitamin D that we make is when cholesterol is converted to vitamin D. So notice that they're very similar. So we start off with cholesterol, we turn it into vitamin D, and that typically happens when our skin is exposed to sunlight. Now that then is a problem. So if you have, so I'll get back to that picture. So we can eat it, all right? But for a lot of people, their vitamin D is not coming from their diet, but rather from their skin. But this can now vary. So if you have light skin and you spend a lot of time in the sun, you have a lot of ultraviolet light, which is going to stimulate a lot of vitamin D. If you have darker skin, The melanin in your skin, it does the good thing of blocking ultraviolet light. That means you are now at a lower risk of skin cancer. So ultraviolet light, it's good that it helps stimulate vitamin D production, but it also causes skin cancer. So that would be at a higher risk in this individual. This individual now has a lower risk of skin cancer, but now they are making less vitamin D in their skin because their melanin is absorbing this ultraviolet light. So they might be at a higher risk to have lower vitamin D levels. And then some individuals just don't spend time in the sun. They spend all day indoors. Maybe they work a night shift. In the past, there were people who worked in mines or children who worked in sweatshops. Some people will cover more of their skin with clothing, which will block exposure to sunlight. If you live far from the equator in winter months, it may be cold. You may not be going outside. So in any case, lots of people have lower than recommended levels of vitamin D. The elderly make less vitamin D in their skin. There's less of a precursor. So there's multiple reasons to have lower than recommended levels of vitamin D. Now, that's a problem. Now, it's a problem for lots of reasons. Vitamin D is actually fascinating. Like other steroid derivatives, it actually binds to a receptor like estrogen and testosterone, which are also derived from cholesterol. Vitamin D binds to a receptor, goes to the nucleus of the cell, and it turns on genes, just the way that estrogen and testosterone does. So there's more than 200 genes whose activity gets turned on. Their transcription is initiated after vitamin D and its receptor bind to the DNA. This affects genes expressed in most tissues of the body. Now, it's famous for affecting calcium absorption and thus the strength of your bones. But these genes, some of them are also involved in inhibiting cancer and also regulating immune pathways and limiting inflammation. And there are a number of lines of evidence indicating that vitamin D deficiencies may have a role in coronavirus. So we notice that vitamin D reduces the cytokine, the pro-inflammatory ones, which are overexpressed in coronavirus infection. Coronavirus infection is affecting some countries farther from the equator, like the United States, to a greater degree than some countries on the equator. Some countries like Japan, where the amount of fish in the diet means that vitamin D levels tend to be higher than in the United States or Europe. Japan has a lower risk of vitamin D. And then also there have been some preliminary studies, once again preliminary, this is early 2021, where if you compare vitamin D levels in patients who have mild symptoms versus severe symptoms, it has been reported that vitamin D is lower in patients where symptoms are severe. And so vitamin D levels may be relevant as well. The last two videos are about how two different tests are done. So the first type of test I'd like to discuss is what's called a molecular test. Now, the next video will be about antibody tests. So antibodies are not testing whether you currently have coronavirus, just whether you've had it in the past. So here, if I've just done a test, this person had coronavirus, but that is not a diagnostic test showing that you have coronavirus now and you are now capable of infecting others. So that's an antibody test, which I'll get to next. You can then do a diagnostic test to test whether you have coronavirus looking for proteins. So we could test, are you currently producing the red spike protein or the blue N protein of coronavirus? And there are tests where you look for the presence of a viral protein. And let's say there could be a color change. If they are. So if there is a diagnostic, this is a diagnostic test, you are diagnosing that someone currently has coronavirus and maybe has the ability to pass it on to others, but you're looking for proteins. The test I'd like to highlight here is looking for RNA, okay? So coronavirus uses RNA to infect cells. So here's coronavirus RNA being injected into the host cell. And one could then look in blood or saliva and then look for this RNA to see if it is being made. And this would then be a sign of a coronavirus infection. So you're diagnosing infection, but instead of using protein, you would then be using RNA. The problem is, how do you get enough to test? Well, once you get a sample from a patient, you can add the enzymes which convert RNA into DNA. And then you can do what's called polymerase chain reaction, or PCR. Essentially what you do is you then take the DNA, you heat the double-stranded molecule to make two separate strands. There are now two separate strands. You then make copies of the single strands to make them double-stranded again, but you only do specific sequences like the virus sequence. So you add a primer, say a primer that only will replicate the viral sequences. So that way, when you start amplifying DNA, you're not amplifying random DNA, you're amplifying the virus sequence. So at first, your patient might not have had enough of the sequence to test positive. So they have coronavirus, but not enough that it would show up in a test. But as you start making more and more copies of this through PCR, ultimately you then get enough that you can test. There are different ways of now testing. One of the ways which is used is using something which would stick to the viral RNA and be fluorescent. All right, so you could test that way. Another advantage of this PCR test is you could, if you wanted to limit costs, you could then test pools. So for example, you could have, these people in pool one, and, there could be 4 people, there could be 20 people in pool one, and then, more people in pool 2, which I'll get to. Let's say nobody in pool 1 has coronavirus. They all provide sample. Let's say, they do a mouth swab, and they all donate their samples into one pool, and one test is done. Let's say that test is then shown to be negative. No other tests are needed. So you've said that no one in pool 1 has coronavirus. So instead of doing 4 tests, you just did 1. If there's 20 people in the pool, instead of doing 20 tests, you now just do 1. That may be a cost-effective way of, say, testing a classroom. So I know at the community college where I teach at Orange County Community College, they were screening patients on those classes which still had to attend in 2020, like say nursing classes that needed hands-on, but they would test pools of students, and they could then test to see, in this pool, nobody has coronavirus. But let's say here's pool number 2, where this person does have coronavirus. Then they do 1 test of the entire pool that would then show that, yes, someone in the pool has coronavirus. And then because they had all of the samples preserved, they could then do then individual tests to find out which individual in the pool had coronavirus. So that is what's called a molecular test, testing for the RNA. And it is a diagnostic test, which would then allow you to diagnose this person currently has coronavirus and is capable of infection. So while there are these direct tests, which would test for, coronavirus infection, and, they could test for the RNA, they could test for coronavirus proteins. There are different ways of directly testing. There's also what's called an indirect test, which is looking for antibodies to coronavirus, sometimes called a serological test. Just pause for a second. In this video, I go into a little more detail, the various controls that one could use in this antibody test. This antibody test is not a test to see whether you have coronavirus, but whether you have had it, whether your body is making antibodies against it. The controls are very important, because obviously you want to be careful of false positives. You don't want to tell people that they are positive for coronavirus if they're not, nor do you want false negatives where you tell someone that they don't have coronavirus if they do. And so would also be a procedural control. You would look for perhaps a color change here to see that the test was working normally. And then once all of the controls suggest that you can have confidence in the results, a negative individual would not have bands for IgM or IgG, where someone who has been exposed to coronavirus could have a test, a band just for IgM, say if this was extremely recent, very early in the infection, where they have not yet developed IgG. Maybe both bands, IgM and IgG, would develop if they say it's within the first month of the infection, so that they've had a recent coronavirus infection. This would be a positive test. If they had the exposure to coronavirus further in the past, they might just develop a band for IgG. So once again, these antibody tests are not testing for coronavirus proteins or RNA. They are looking for the human antibodies made after exposure to coronavirus here. And once again, the reason to test for these two antibodies, IgM and IgG, is that once again, they can follow a different time course as far as their production. IgM peaks earlier as far as its production, whereas IgG production can go on for months afterwards. So this was a video summarizing some of the latest understanding of what coronavirus does inside human cells and interacting with various components of the human immune system, with some preliminary information on the first coronavirus vaccines and two different types of coronavirus test.